So one of the joys of being a professor is to learn about the world through our students. And I think I learned something about India when British once told me that every morning in school they recited the poem Mind Without Fear by Tepper. And I'm afraid that my Bengali is a little rusty, so I'll read it to you in English, the translation by Yates. So, Mind Without Fear. Where the mind is without fear, and the head is held high, where knowledge is free, where the world has not been broken up into fragments by narrow domestic walls, where words come out from the depth of truth, where tireless striving stretches its arms towards perfection, where the clear stream of reason has not lost its way into the dreary desert sand of dead habit, where the mind is led forward by thee into ever-widening thought and action, into that heaven of freedom, my Father, let my country awake. So that's a little bit different than the Pledge of Allegiance. Now, I'm not claiming a causal relationship here, but Greenidge's mind is certainly without fear, and he's undaunted by walls that would limit his knowledge. He is, we left India for a master's in biomedical engineering at NUT, and he almost literally, I think, clambered over their walls to come to our neuroscience program. And even as a BNS student, he also pursued courses at Columbia, workshops at NGH, and the summer school at Cold Spring Harbor just last summer. But to really see his tireless striving, I think you have to look at his devotion to research. He started the program in January 2010, so the work we'll present today was completed in exactly 5 years. And looking back over the emails we exchanged at that time, it's actually fairly remarkable that he has anything to present at all today. Because there was no project when he started. He may not have known this, but I knew there was nothing. I told him that I'd become interested in transcranial stimulation, and I'd like to see if we could use it in our research. And this is almost the exact opposite of what the NIH thinks is a good research project, right? There was no hypothesis, there was no grand plan, there was just the thought, wow, this is awesome, let's do it. And so we started on this big adventure, but without Greenidge's tireless striving, none of this would have happened. He searched for the equipment, he soldered it together, he programmed it, he developed paradigms, and he kept on widening his thoughts and actions to find the truth about transcranial stimulation. He did behavioral studies, functional imaging, single-cell recordings, multi-electrode array recordings, network models, and compartmental models. His work is certainly no dreary desert of dead habit. Although personally I'm a little skeptical that this will lead to a heaven of freedom, but we're very fortunate to have walked a few steps towards the depths of truth together with Greenidge. And I hope that we will continue our search for that elusive, clear stream of reason together. But before we go there, you have a job to finish. So in the next 45 minutes, he'll take you on a whirlwind tour of the mechanisms of transcranial electric stimulation. Thank you.
Thanks, Bart, for this great introduction. And I don't know if I can live up to that, but I will try my best. So today I will be telling you all about the mechanisms of transcranial electrical stimulation. So electricity has been used in therapy for a very long time. Actually since AD 46, where Roman physician Scribonius Largus used this live torpedo fish to treat headaches. Just put this live torpedo fish on the forehead and you'll have some relief from pain. Since then it has been used for treating toothache, gout, and so on and so forth. And in the early 20th century, it also started being used as electroconvulsive therapy. And it was pretty effective, and it started to be known as the penicillin of psychiatry, and of course immortalized on screen by Jack Nicholson. But the image today is not so gruesome, and we use a slightly subdued version of this technique called transcranial electrical stimulation. And the currents are almost 100 times lower than ECT. So I am studying this, and I'm going to talk all about this today. But why is it important to study? So let's look at this. Why is it important? Why should we at all care about the mechanisms of TES? So transcranial electrical stimulation has been implicated to aid different kinds of treatment of different neurological disorders. Depression, Parkinson's disease, chronic pain, schizophrenia, stroke recovery, epilepsy. And not only all the big players in neurodegenerative diseases, it is also implicated in changing your lucid dreams during sleeping, or memory consolidation during sleeping. And if you think this is like the entire picture, you're mistaken. because it's just part of the whole story. It has been implicated in multiple other domains. So looking at this, you can start thinking, at least I start thinking that maybe it's too good to be true, all these effects. But imagine even if 10% of these effects are real, all you need is a 9-volt battery and a pair of sponges to implement this technique. So it's a really handy technique, and therefore it's important to study the mechanisms of transcranial electrical stimulation. All right, so what we have here is, the way we set up, our setup works is that we place 2 big sponge electrodes across the head and we pass current. We connect it to the current stimulator and we pass current through it. The current is of around 1 milliamps of amplitude. There are generally 2 modes of application of transcranial stimulation, at least in current practice. One is TDCS, the transcranial direct current stimulation, where a constant current is passed. One electrode is positive and the other is negative. And the other one is TACS, where alternating current stimulation, and we can manipulate the frequencies and amplitude and so on and so forth. So today my talk consists, I'll only focus on TACS from now on. Right? So I'm going to only focus on the mechanisms of TACS. And when I say mechanisms, well, mechanisms can be described at multiple levels. And throughout my thesis, I have tried to at least look at have a multi-level approach. So, for example, aim 1 and 2, I have looked at behavior and then predicted what could happen synaptically in molecules and neurons and networks. And then I did recordings in vivo in awake macaques, trying to test some of the hypotheses that we developed from behavior. And in my future work, I would like to address some of the system and map level mechanisms. So before I give you what I found, I want to tell you what is briefly known about TACS mechanism. So one thing is pretty clear, that if you have an alternating current like that, with a positive cycle and a negative cycle, the effect it has on a neuron will also flip based on what cycle it is. So that's pretty much taken for granted. Okay? And when it depolarizes or hyperpolarizes based on the field, the membrane polarization caused by a current of around 1 milliamp is 0.15 millivolt, which is pretty low, right? And this has come off from some in vitro studies and some modeling work. And for isolated cells, the membrane polarization reduces with frequency. So if you look at how much effect there is on membrane polarization with different frequencies, you see that it drops off with increasing frequency. So when we are studying TACS, seems like this is kind of a nice frequency to choose. So all my experiments that I will show, I've chosen 10 Hertz as my chosen frequency for applying TACS. One other thing that is really interesting is this idea that this alternating current can entrain a network of neurons. This is some work done by Frolic and McCormick in 2010. They showed that if you have, this was in ferret brains in vitro, so you have some sort of bursty activity here that happens like this with no applied electric field. When you apply an electric field of 4 millivolts per millimeter, a sinusoidal field, you see that this activity almost gets entrained by this applied sinusoidal field. All right? And you can see that if I project it back when there was no stimulation, this is not lined up with the peak of the stimulation. And very similar results has been shown in vivo by work done actually in this department by Simol, was then working with Yuri Gujaki. She recorded from hippocampal CA1 neurons. So here it's plot from one of her papers where the state of stimulation and different units that was recorded, and the red, the color is the activity. So you see the activity kind of peaks at the peak of stimulation. So this is a very interesting possibility. If you can entrain neurons like this in the brain, that would be something really, really promising as a neuromodulatory tool. But there are a few problems, there are a few concerns. So when we hook up the subject like this, before the current can actually reach the brain, it has to pass a lot of hurdles. And those hurdles come in the form of skin, fat, muscles, skull, cerebrospinal fluid, and they have pretty high conductance. So modeling studies suggest that half of the current that you apply is shunted around this part, and then remaining 7/8 gets shunted in the CSF. So what's left is a very low current. And electric fields, so here I'm showing that most of the current that you apply gets dissipated into the skull or skin and fat, and very little comes into the brain. And estimated field strengths are around 0.11 millivolts per millimeter, which is actually even lower than what Simol had applied. So the question really is that can this very small electric field do anything to the brain? It does something very interesting actually. So when you hook up a subject like this, you put one electrode at the back of the head in the occipital cortex, and one electrode at the top of the head in the vertex, and you connect the subject to the stimulator and switch on the stimulator at 10 hertz and 0.5 milliamps. What you see is, you see some peripheral flickers like that. And that's phosphines. So you're stimulating, is very, it suggests that you're stimulating V1 and you see this visual stimulus come up, and the field was very excited. The field of TACS was very excited. And there was some earlier work done by Kanai and colleagues, and they found something very interesting as well. What they found is that these are different TACS frequencies applied, and this is the rating of phosphines. So how strong are the phosphines that is generated at 1 milliamps? They saw that the phosphines generated are strongest at around beta frequencies. Okay, and this is when the stimulation was applied with the lights on in a room. What they noticed was that when you switch the light off and when it becomes dark, this peak frequency actually shifts towards alpha. And their explanation was that during light, the dominant EEG frequencies are in the beta range, and during darkness, the dominant EEG frequencies are in the alpha range. So maybe TACS is sort of syncing up with the ongoing oscillations in the brain, and therefore this is happening. That's a really good interpretation, or a very promising interpretation for TACS and entrainment, but there might be another story here. So what they suggest is a model like this, where you apply stimulation and the current just locally excites a particular area underneath the electrode. And you have occipital stimulation, and it's pretty focal, and you get phosphines. And this almost suggests that with stimulation you can induce spikes, and it's a suprathreshold stimulation. All right, and we know that if you make a V1 neuron spike, you will see phosphines. So if you can make a neuron spike with V1 neuron spike, you will get phosphines. But there is a slightly other explanation to this phosphines too, and that has also been suggested in the literature, is that when you stimulate like this, maybe the current just spreads to the entire skull and reaches the retina, and that's what causes the phosphines. So this would slightly be a different mechanism, because then we are having retinal phosphines, and probably the current that goes into the cortex underneath the electrode is very subthreshold, very low, and that kind of matches up with the predictions from the head models. So we went ahead and tested it. How did we test it? So we had a subject, and we put the reference electrode on the vertex, and we put one electrode at the back of the head and measured the phosphine threshold. So here I've got a phosphine threshold, which means the minimum amount of current to generate a phosphine. And this is different frequencies. So let's just look at 20 hertz. So the hypothesis is that if the source of phosphines is in the retina, as you move closer and closer with the electrode towards the retina, your phosphine thresholds should decrease. That means your phosphine, the current intensity required to induce a phosphine should go down. All right? So let's see what happens. It went down when we moved from OZ to T5. Even forward to the frontal lobe, it went further down. And it is consistent across different frequencies. And we used a retinal ganglion cell model to fit this data. And you can see that this also looks like a U-shaped curve. And this preference of alpha in this tuning is also there. And so the idea is that this tuning for alpha can just come from a property of retinal ganglion cell. It doesn't need to be entrainment with a different brain oscillations. And this work has been further validated by a modeling paper done independently by Latso and Hirata in 2013. They took our data and our placements and predicted what the current densities should be around the skull. And they also claim that the maximum current intensities are around the eye, when you apply stimulation like this. So that suggests that stimulation is inducing phosphines at the retina. We also did another experiment where we tested another hypothesis that would again bring us back to whether stimulation induces phosphine in the retina or in the cortex. So the idea is pretty simple here. So if you put an electrode at the frontal lobe, very near the eye, it's known that it induces phosphines at the retina. So the claim is that if the phosphines are occipital, if the occipital, when you stimulate the occipital cortex, if the phosphines are generated in the occipital lobe, the condition would be like this. So the focal prediction is that if you stimulate frontal lobe, it will take around 60 milliseconds to reach the cortex, the information to be transmitted to the cortex and for a percept to happen. But if you stimulate the occipital lobe, you will almost immediately should get a percept. Or the information should transmit immediately to the cortex. The non-focal prediction is that both generate in the eye, so they will both have a delay. And I'm not going to go into the data, but we found our results are consistent with this non-focal prediction. All right, so to summarize, I've shown you that stimulation is pretty non-focal, and the currents spread well beyond the area of stimulation. And so most of the current is basically shunted through the skull, and very little current reaches the brain. And we are interested, so I was interested further in knowing what can this current do to the brain. Does it do anything at all? One thing we can predict from these experiments is that the effect is sub-threshold here. That's one thing. All right, so this is the question that led me into my aim 2, that can this small current induce any behavioral effect cortically? All right, so before I go into M2, I have to pick a brain area to study TSES. So I want to see if it can induce something cortically, but the cortex is a very big place, and some people might believe that the whole cortex is doing vision, but some part of the area we know for sure is doing vision. And I chose to stick to the visual areas for a few reasons. One is that it's pretty well explored functionally, or relatively well explored functionally and anatomically. We know that certain areas code for certain stimulus features. And there are objective measures to quantify behavior. And also there are homologous areas in the macaque where if you find something behaviorally, we can go in and test. That's, again, this is still a very big area. So we zoomed in more. So I specifically exploited the visual motion detection system, area MT, area HMT plus in humans and area MT in macaque. Two features of this area are of interest for this talk. One is that this area is sensitive to motion direction. Neurons in this area code for motion direction. And the other is that this area is lateralized. So if you look at the center and so I present something on the right visual field, your left MT is going to encode it. Same for the left visual field. Now, the task. It's a very simple motion direction discrimination task. Let's say I show you this dot pattern that is going upward, and I ask you, is the dots going up or down? You're going to say up, right? And so I'm plotting here motion strength, the coherence, which in this case is 100%. So I define positive coherence as upward moving dot and negative coherence as downward moving dot. So 100% here means all dots are moving upward. And on the y-axis, I'm plotting the proportion of upward choice. All right, so for this stimulus, it could be 1. If all the stimulus are moving randomly, all the dots are moving randomly, you will be at chance of saying up or down. If all the dots are moving down, you basically always say down, so your proportion of upward choice is 0. If you repeat it across the spectrum of motion coherence, you get something like this, and then you fit, generally we fit a cumulative Gaussian, and this function is called psychometric function. So this is an objective way of quantifying motion direction discrimination in human subjects. Again, there are two features of this particular graph that I'm interested in. One is the point of subjective equality, which is at what coherence is the subject equally likely to say up or down. And for an ideal subject, this should be at 0% coherence when the dots are just moving randomly. The other feature is the slope of the graph at the point of subjective equality. The slope is just some del y by delta x. So a more intuitive understanding is that if the sensitivity of the subject increases, for example, if the graph becomes steeper, for the same change of y, now you need a smaller increment in your motion strength. So the subject has become more sensitive, right? So with this, I'll present to you the data from my first experiment. So what we did, we have this task, we are going to stimulate the subject over area MT, left MT, and see what happens to this behavior. And remember that stimulation here is done at 1 milliamp 10 hertz, 0.5 milliamp 10 hertz, and it has no information about motion direction or anything. So first is just a baseline graph of performance without stimulation. So let's see what happens, this data from one sample subject. When we stimulate, we saw this increment in sensitivity. And this was consistent across the population. It's really surprising because stimulation has no information on motion direction. And we see this across the subjects. And if we do a control experiment with ipsilateral stimulation, that is, we stimulate the left MT, but now also show the stimulus on the left visual field. So this is not the relevant visual field for that particular, for this MT, for the left MT. We don't see the effect. It's pretty inconsistent across subjects. So you might be tempted to think that stimulation is adding information to the system, but it doesn't have any information about what motion I'm presenting. So another way of thinking of improving sensitivity could be maybe it reduces or hampers something that was making you become less sensitive for some reason. There is some mechanism like that in the cortex. So adaptation, for example, whenever you present a stimulus for a prolonged period of time, you get adapted to the stimulus, and that adaptation reduces your sensitivity during these kind of tasks. It has been shown before. So our hypothesis was that maybe stimulation is interacting with adaptive processes and reducing adaptation, thereby increasing sensitivity. Just to give you a brief explanation of what adaptation is and what kind of after effect it induces, I will let you look at this nice video here. So just focus at the center of the screen for a long time, all right? 40 seconds. Keep focusing. What's happening is that the outer spirals are going inward and the inner spirals are going outward. So this is a prolonged presentation of a very particular type of motion. So if you keep looking at the center of the screen for a very long time, you will see what it does to a very static stimulus presented consequently. So just keep focusing at the center. Try not to move your eyes. And your neurons have started to get fatigued. So feel at one with the stimulus, as if it's do you see the sky move?
Yeah.
All right, and I can show you later that this is not an animation in the video here. I can do it. You can also look at the back of your hand and see it move, but maybe the effect is gone by now. But this is a nice, after effect is also known as a waterfall illusion. You can look at a waterfall and the same thing happens. If you look at your car, it will start going upward maybe. But vision scientists are, we are boring people, so we don't like this kind of stimulus. Instead, we use some very nice, interesting stimulus, the dots. So we come back to dots again. And so the adapter now is not this nice stimulus, it's a bunch of dots moving upward. And we adapt the subject to this upward motion, and the expectation is that on a subsequent presentation of a test stimulus that is moving randomly, the subject is now more likely to say down than up, right? So what's the effect on the psychometric function? The effect is pretty simple, that this point should just go down, and if you trace the whole spectrum, then the graph is going to shift. So there are two main effects that have been reported in literature. One is that the PSC shifts to the right, and the other one is that the slope reduces. The slope is a sensitivity. And so we did this experiment in the human subjects. So we had upward moving dot as adapter, and then followed by the same test stimulus that we were showing before. Right, so this is again no adaptation. So with adaptation of upward moving dot, we see this is data from one sample subject. Again, this shift in psychometric function. And if you look closely, there will be a shift in the sensitivity. There is one. So the question is, what happens when you stimulate during this time? Consistent with our prediction, what we found is that stimulation reduced adaptation. And it not only reduced the PSEs, it also undid the change in sensitivity. So first of all, this reduction is consistent across the population. Second, this effect, this reduction of MAE is directly proportional to the amount of MAE induced in the first place. Better explanation is here in this graph, and I will explain this graph a little bit more elaborately now. So if you look at the psychometric functions that I just plotted before, so what you are seeing in the x-axis here is the difference between the PSE of the black and the blue. That's plotted in the x-axis. So that is the strength of the motion after effect. What is plotted in the y-axis here is the shift back from the blue to the red. So what I show you here is basically that if you have a lot of motion after effect, you're going to have a lot of reduction of motion after effect. So the efficacy of TACS is dependent on how much motion after effect there was to begin with. So it's sort of a brain state dependent mechanism. Same thing happens with sensitivity. What again I'm plotting, I'm comparing the slopes of these graphs. So on the x-axis is the difference between the black and the blue, and on the y-axis I plot the difference between the red and the blue. And we again see this negative correlation. So stimulation is undoing the effects of adaptation, and it is proportional to the amount of adaptation that is induced. All right. So we have some mechanism to think about now, adaptation. So did I answer this question? Can the small TSCS current induce any behavioral effect cortically? Yes, it can. And what happens with stimulation of HMT plus is that TSCS increases motion sensitivity, it reduces motion after effect, and this effect is proportional to the strength of the motion after effect. So the question that I followed up with is, okay, and this is cortically induced because this only happens for the stimulated hemisphere. It doesn't happen for the non-stimulated hemisphere. The question that I followed it up with is, how does TSCS affect neurons in area MT to induce this behavioral effect? And again, the hypothesis is that it reduces motion adaptation. So anything that happens in the neurons with motion adaptation should change or should be attenuated with stimulation. So that's our hypothesis going into area MT, and area MT of the macaque. Unfortunately, we can't record from humans. Strength of adaptation also determine efficacy of TSCS. So this brings me to my aim 3, where I test these hypotheses in area MT of the macaques. This is our setup. The monkey looking at fixating a dot in the center of the screen. We place the electrodes around the recording chamber, slightly different than what's done for the humans. Technical limitation, we can't, because we have a chamber from where we record area MT neurons, so we can't put the electrodes right on top of that area. So we have to live with this limitation for the time being. The first step is to locate the receptive field of the neurons. Generally, it's located on the opposite hemifield. So I'm recording from the left MT, I will see receptive fields on the right hemifield. Locate the receptive field, and from there onward, put the stimulus on the receptive field of the cell. 4 conditions that I have tested. The 4 conditions are no adaptation, yes adaptation, and no stimulation and stimulation. So I'm going to explain this paradigm a little more. I'm going to elaborate on it. So let's take the first one. No adaptation, no stimulation. All right? What we do is, first, for 3 seconds, the monkey is fixating on this dot, and for the first 3 seconds, I just show randomly moving dots, right? So I'm not adapting that neuron to any particular direction. And then I test it with coherently moving dot at 8 possible directions. So that gives me the tuning curve of the cell. So let's say I first showed a downward moving dot pattern for 0.3 seconds. In the next trial, I have again random and then test some other direction. So if I map out again the whole direction spectrum of the test stimulus, for MT neurons, as I said earlier, you see that it prefers a particular direction. In this case, this is a cartoon I chose upward moving dots. Okay, so this neuron prefers upward moving dots. You can feed a von Mises function to this, and that we call that the tuning function for this neuron. So now I know that this neuron prefers upward moving dots, so I can specifically adapt this neuron to upward moving dots, right? So that's my adaptation without stimulation condition. So same thing, instead of the random, now I present coherent adapter, which is dot moving in the preferred direction of the cell for 3 seconds, followed by the test stimulus. And the typical result is that the neurons reduce their tuning amplitude, and it's a cartoon, but they also reduce the tuning width on adaptation. And this data has been shown by Colin Mofshan in 2003 and has been replicated since. My main question is that what happens to this blue graph and this black graph when I stimulate? So basically I stimulate here and test again, or I stimulate during this time and test again. And I'm interested in what happens in these two conditions. And as you know, I have specific hypothesis for this. So let's see a sample neuron, what it does. Again, 4 conditions. So the first one is just baseline tuning of the neuron. Adapted to its preferred direction, the amplitude drops. So I avoided the data, the real data points here to avoid some cluttering issues of the images, but the fits are pretty good. You have to trust me on this. You see that this amplitude reduces. So I'm looking specifically at what happens to the tuning amplitude with adaptation and stimulation. So now I stimulate while I have the adapter. All right? That's exactly what we predicted. It's pretty consistent with our behavioral results. It pushes it back up to the unadapted condition. What happens when you stimulate the no adaptations? Not much. This is a very standard result of adaptation reducing tuning amplitude. We also found many cells that does exactly the opposite. So when you don't adapt, we have a baseline tuning graph, and when we adapt, it actually increases tuning amplitude. This is pretty interesting by itself, but what I'm interested for this project is what stimulation does to this graph. What we found is that if adaptation pushes it up, stimulation is going to pull it back down, which is again consistent with the idea that stimulation is undoing whatever adaptation is doing to the system. Again, without adaptation and stimulation, not much of a change. So TSCS attenuates adaptation-induced facilitation and attenuates adaptation-induced suppression. And recorded 107 cells across 2 monkeys, and the result is pretty consistent. So in this graph, what I'm plotting is this quadrant is the first case. So on the x-axis is the blue arrow, on the y-axis is the red arrow. You see that the directions are opposite, which shows that there's a negative correlation or negative relationship. And this quadrant contains the cell that I have shown in the second case, where adaptation actually increases facilitation. So it's negative, positive, and we see that across population, we have a negative correlation of the effect of adaptation and the effect of stimulation on adaptation. So pretty consistent across population of tuning amplitude results. What about tuning width? Again, pretty similar story. If adaptation increases or broadens the tuning width, stimulation will sharpen it. Okay? If adaptation sharpens the tuning width, stimulation is going to broaden it. Of course, we always show the best neurons around here, but it is consistent across population again. So this is the same plot as before, just showing that this data is across the population. So if adaptation reduces tuning width, stimulation undoes that reduction. Right? So we didn't not only look at neuronal tuning properties, we also then further looked at changes in evoked local field potentials. So again, let me remind you of the paradigm here. So I present the adapter or the random stimulus for the first 3 seconds and then present the test stimulus. So here is one sample recording session, recording trial. You see that this is when there was no stimulation. So there is some evoked response in the beginning of the adapter or random, and then there is evoked response at the test stimulus. When you stimulate, we have all these artifacts of stimulation during this time. So I pretty much only analyze data around this time point. So everything before the test stimulus was discarded for further analysis. So I'm going to show you what happens to the evoked local field potentials when with and without adaptation, with and without stimulation. So first condition again is baseline. You put a stimulus here that's moving in a particular direction. I'm only showing you data that has been averaged over the direction that is closest to the adapted direction. So this graph is the baseline evoked local field potential. When we adapt, I don't see much of a reduction in this evoked field response. And when I stimulate, I see a tremendous increase of the amplitude of this evoked response. And this is the same for stimulation with adaptation, stimulation without adaptation. So this tells me that stimulation is overall increasing the responsivity of that area. And this is not specific to adaptation. This is the only effect that I'm going to show you today that is not specific to adaptation. Still thinking about the probable mechanisms of this. Of course, you have to first understand what LFP means to actually understand what this means, but we'll be there very soon. Another analysis that I did was a spectral analysis of the local field potentials. So here I'm plotting LFP power on the y-axis and for different frequencies. This is normalized power. So the first graph is for no adaptation. When we adapt, we see a decrease in power across the spectrum, more so in the gamma band. With stimulation, again, overall broadband increase in power across the spectrum. It is not specific to 10 Hertz. We stimulated at 10 Hertz. There's nothing specific here about 10 Hertz. All the power across the spectrum just increases. And with stimulation and no adaptation, again, the increase is not clear and it's not significant. So this is again adaptation-specific increment, broadband increment in power. So I come to the end of my aim 3, and I started with the question, how does TSCS affect neurons in area NT? I have a few answers for that. First, TSCS attenuates adaptation-induced changes in neuronal tuning and local field potentials. I've shown you that. TSCS effect size depends on adaptation, and it induces adaptation-specific broadband increase in LFP power. So the data actually is pretty consistent with the behavioral finding. So maybe what TSCS is doing is interacting with the adaptive processes in the brain and hampering it, attenuating it. We still don't know whether it reduces adaptation altogether or reduces the speed of induction of adaptation mechanisms. There are more detailed questions that needs to be answered, but one thing to pinpoint is adaptation is getting affected. And of course, no evidence of prolonged frequency-specific entrainment. So it might entrain the brain areas while we are stimulating, but once stimulation is off, there is no prolonged frequency-specific entrainment. There has been some data showing that there is prolonged frequency-specific entrainment from other labs, but for us, we didn't find anything specific to the frequency that we applied. So most of this work is empirical data. Let me speculate a little bit now about what might be causing this, how adaptation can be reduced. There is some data from McCormick's lab, recordings done by scientist Vives 2000. They did recordings in the CAD-V1, and they found that this long-lasting adaptation in CAD-V1 is mediated by some potassium channels. So their results show that in V1 you have sodium channels, delayed rectifier potassium channels, the high threshold calcium channels, but also as a negative feedback mechanism you have this sodium dependent and calcium dependent potassium channels that are outward channel, hyperpolarizes the cell as the cell starts to spike, and that's the mechanism that mediates adaptation. So one of the proposed mechanisms, according to me, is that maybe stimulation is interacting with the mechanisms of these channels. So I did a simulation study. Basically, I just implemented this model this is a Hodgkin-Huxley type conductance-based model. And I implemented this in MATLAB, and I just added a sinusoidal stimulation to the membrane dynamics. So again, I tried to keep it as close to the behavioral and the electrophysiology paradigm as possible. So I will inject a current for 4 seconds, and then give it a little bit of gap, and then inject the same current again for 1 second. And I'm going to test what happens when you, so this is sort of the adapter, and this is the test. All right, so this is data from the cell without any stimulation, just the simulation of the cell. You can see that it's very difficult to see properly what exactly is going on, but you can see that the cell is spiking very highly here, and then slowly it adapts. And then for the 1 second test stimulus, the response to begin with is not as strong as it started here. So the cell definitely has adapted. What happens when we stimulate this cell at 10 Hertz? That's what happens. So again, not clear from looking at this graph, but you can kind of start seeing this entrainment to the oscillatory membrane fluctuations. So that's one thing. The other thing is to look closely at this window, very starting point. You'll see that the red and black curves are not overlapping. They're slightly different. Even more close, or a better way to represent this data is to look at the interspike interval across the test time. So this is the onset of the test stimulus at 5,000 milliseconds, goes till 6,000. So when TSES is off, you see that the interspike intervals are much larger, which means that there was a bigger adaptation. And if TSES is on, the interspike intervals are smaller. That means the cell is spiking more, which means that it wasn't adapted as much here. So I'm not saying that this is the only mechanism that can explain my result, but this is one possible mechanism and a pretty easily testable mechanism in vitro that can lead to some more understanding of the mechanisms of TSES. So my final slide, or semi-final slide, conclusions, mechanism of TSES. I have looked at multiple levels of descriptions of mechanisms. So from behavior, I've seen that TSES induces retinophosphines, which has made me conclude that TSES is a non-focal stimulation technique, probably sub-threshold stimulation. It increases motion discrimination sensitivity and reduction in motion after effect. It reduces motion after effect. So this hints to the mechanism of attenuating adaptation with stimulation. I looked at neurons in area MT. Again, attenuation of adaptation induced changes in neural tuning curves. In terms of network, increase in overall excitability, and adaptation-specific broadband increase in LFP power. And taken together, this actually again confirms this attenuation of adaptation mechanism that was concluded from the behavioral studies. We also found some adaptation non-specific effects, that is increase in overall excitability. Speculations? Maybe the membrane fluctuations are interacting with potassium and the sodium-dependent and calcium-dependent potassium channels. Future directions, I want to study more about this system and the mechanisms of TSES, and more now in terms of maps and systems. So we have already been running experiments in Rubik with concurrent TSES and fMRI. We collected a lot of data, and preliminary results look pretty promising, and actually we do see somewhat of an evidence of reduction in motion after effect, but that's very preliminary analysis. We are doing multi-electrode recordings in V1, and the idea is that adaptation is not specific to MT, adaptation also happens in V1. So if this is a very general mechanism, if I can reduce adaptation in MT, I might be able to reduce adaptation in V1 as well. Some advantages of doing these recordings in V1 that we chronically implant arrays, so now we have more time and I can actually look at multiple frequencies, the effect of multiple frequencies on, multiple TSES frequencies on adaptation. So we will be using gratings instead of moving dots for V1. Yeah, that's it. And the Oscar slide, I'm going to thank people. I always felt that whenever I saw people presenting theses and they get really emotional at the end and say like, I always used to sit in the audience and feel that that's not going to happen to me. But even when I was writing the acknowledgement section of my thesis report, I started feeling really sad. It's terrible. I don't know what to do with that. But I am really grateful to a lot of people for helping me out throughout my PhD. First, people who funded me, definitely Charles and Johanna Bush Foundation from the beginning, and then National Eye Institute for funding me. My committee members, Denis, Jim, Farzan, Marc, who came from Duke. Thank you all for being part of my committee, and I really appreciate your input to my thesis work. And last but not the least, Bart. I don't think I could have had a better experience in a lab, and I think I want to keep it as short as that. And everything that followed, what has happened over the last 5 years, I'm really thankful to Bart the way it turned out, and it's absolutely amazing. I was such a different person in 2010. Worse. I have changed a lot, and Bart and all the lab members have helped shape me in many, many positive ways. The entire CMBN faculty, it's been a pleasure being in this department, learning so much from all of you through courses, through discussions during talks. One of the things that I enjoyed the most was the question asked after the talks. So that is something was really good, because I would sit in the audience just thinking like, I didn't understand that part, I didn't understand that part of the talk, and then somebody from our faculty would ask this very nice question, and all of a sudden everything, I would be able to join the dots. So definitely for that, and all the lectures and everything. And everybody is just, I think, very friendly around here. And I've been to a couple of other departments, and I have other experiences there. The staff, Connie, Anne, Wayne, Selena, Shiva, the OISS, the Office of International Students, and the entire CMBN animal facility. Very, very helpful to me throughout my entire PhD. And all past and present K-Lab members and all my friends in the CMBN and Rubik, they have all been very, very influential in shaping me as a person, as a scientist. And I'm very grateful to them. I would just like to share a few memories with you, and I think these are the memories that I will miss when I leave this place. I got to be in a celebrity magazine for growing a mustache in the first year. That's great. I just came, everybody had a mustache, I had one too. And we had great celebratory send-off for the labs. So this is Fred getting his farewell party. So that was really interesting that the PI lives the students. In India, if you have been studied in India, this is, you can't imagine this thing. Again, coming back to Bart, there have been many days when I went into his office with crazy ideas, and a lot of times the crazy ideas have been criticized and rejected, but sometimes even crazier ideas have been accepted, and I have been allowed to do experiments. And one such crazy idea was doing this experiment with a conductive mask around my eyes and try to see if I can sort of eliminate phosphates. Looked pretty crazy at that point. One more is I'm really thankful to the physical plants here, because I think they have played a very big part in my PhD. Because how to deal with non-favorable conditions, how to survive and thrive in these conditions, I have learned from them. Because in winter it is hot here, and in the summer it is cold. I've made ingenious ways of coping with summer cold. One thing I'll miss really a lot is also the coffee made in our lab. It's really, really awesome coffee. If you haven't tasted it, you should. Thank you.
Time for questions? I have just one starter question. It's a small technical question. Remind me, how did you measure the phosphine?
The phosphine thresholds?
Yeah.
So it was a 2 alternate force choice task. So the subject was sitting and there was a beep in the middle of 2 intervals, and I either stimulated in the first or the second, and I asked which interval did you see the phosphines, and I changed the intensity of the phosphines in an adaptive staircase way to get to the threshold detectability.
My other questions are very theoretical. I take your point from your third specific aim that you saw no effect of prolonged stimulation.
Yes.
And in one sitting.
You mean?
So in other words, with a subject, you're just, so those are primates, so those animals are they're getting repeated stimulation over
Yeah.
days,
Yeah.
as well as shorter or longer. Did you ever look over days to see if there were cumulative?
That's a very good question, actually. So not too many studies have looked into multiple days, but it is also difficult to do because
Sure.
the cell that I record for a day, I don't get it the next day.
Sure.
So any differences between the 2 setups can be attributed either to the cell or to stimulation. So that's one thing. And so again, what I found is that if you stimulate for 3 seconds, the time that follows stimulation, I don't see any effects that leaks into that 0.3 seconds. Now, maybe if I stimulate it for 30 seconds, it might happen. And many earlier studies or human studies, they have actually looked into this longer stimulation periods, and they did find prolonged effects of stimulation. It's just, I think, difficult to do with monkeys where they have to fixate and I have to control for some of these measures.
Okay, so actually my question was back to the human condition. You could argue that someone who has used a drug of abuse for a long time has adapted their brain. And I'm wondering if you think there's any chance that such a stimulation paradigm would be useful in therapeutic treatment. And I guess it wouldn't be good to take a brain that was, on drugs and had reached the point where they're a heavy user, because then you're going to take them back up to using even more.
Yeah.
But someplace in the curve where they become the abusing brain. Is that all just pie in the sky?
No, there are some studies. So there are kind of 2 branches of doing this kind of research. So one is looking at mechanisms, and the other is just applying it in the clinic and just seeing immediate results. And one inspires the others and so on. So there are some studies that have looked into changes in addictive behavior with transcranial direct current stimulation, and they do see some positive effects of whatever paradigm they're using. But the effects are not reliable, and it only happens for a certain percentage of the population that they have tested. So the idea is that if we can pin down the mechanism, then it will optimize the use of the TDCS in this ways. But also I think that maybe one thing I should make clear is that the adaptation of the brain or the long-term changes of the brain that happens during addiction is sort of different than what I'm studying here, right? Because I'm just looking at these very short-term adaptive changes. I don't know the answer, but there are definitely a lot of labs interested in studying addiction with stimulation.
That's a simple question. When you did the phosphine origin studies, did you have a chance to focus on the LGN? Because most of the cases you probably affect.
At least from the modeling studies, the effect in the very superficial layers of the cortex, the electric fields are around 0.11 volts per meter, millivolts per millimeter. It's really low, and the predictions are that there is going to be almost very negligible, no effect on LGN. And behaviorally, I don't have a very good way of saying whether it's V1 or LGN. As much as I do sort of have an advantage saying V1 versus retina. But in the cortex, if it originated somewhere in the cortex, I had no idea where it could originate. For example, even in the adaptation study, I have stimulated MT, but V1 has direction-specific cells, and I don't know whether these are originating in V1. I just see the effects in MT, which is consistent.
I have kind of a similar question to Joan. There have been a number of studies that have shown that you can actually alter the responses of people with schizophrenia or depression by using this type of stimulation. And one wonders then if you're actually changing networks in a way that you're not capturing here. And is there a way that you could look at those sorts of sort of semi-permanent changes that happen across time? I mean, you've got dense array EEG information. Can you do directed coherence or modeling of networks by looking and seeing whether you're actually changing things across time?
Definitely, and actually there are some other labs doing this exact same kind of studies where they use EEG simultaneously with TACS and TDCS and look at some of these changes. And I think that I have to make a decision of what measure I want to look at. And I'm sure that the interpretation of the data with EEG and this cross-coherence spectrums, these are not that clear either. And there are many things. For example, like if you look at my LFP data, somebody would argue that there is tuning in gamma band, so there's information about the stimulus in the gamma band, and LFP is a combination of the input that is coming to an area. To me, LFP is just the signal that we have left after filtering from 0 to 120. And really, we don't have very good idea where these are originating. And so for personally, I wanted to focus on something that we know exactly what's going on. But for example, like schizophrenia, people think that this is a computational problem in the brain. It's a network level effect, and maybe stimulating is going to have an overall network effect that is going to elevate some of the symptoms. And actually, at least according to the papers, it is true. So definitely there is more scope, but I think the more good grip we have on the actual mechanism, it's better. It's a better step forward.
But also looking not just while you're stimulating, but looking at in the future, like having them come back 2 months later and looking at whether they actually have some differences in the way that they're responding to the stimulant from the control group that has not had that sort of stimulation.
These are very interesting studies, and we will have to do them in order to address some of these issues, definitely. Yeah.
Other question? Right. Thank you.